Good morning, students. Today, let us learn about law of independent assortment or Mendel's second law, dihybrid test cross, and incomplete dominance. First, Mendel's second law. It is also known as the law of independent assortment. After a careful study of the results of a number of experiments on the inheritance of two characters at a time, Mendel expressed his conclusions in the form of another law, the law of independent assortment. It is generally known as Mendel's second law. He found that the factors that determine two characters assort independent of each other during gamete formation. Hence, he proposed the theory of independent assortment. It states that two pairs of factors or alleles that determine two characters assort independently during gamete formation. I repeat the same, two pairs of factors or alleles that determine two characters assort independently during gamete formation. Mendel collected evidence for his theory by the study of seed color and seed shape. This law is the outcome of Mendel's dihybrid experiment. Next, explanation for Mendel's second law. In garden P, yellow seed color is dominant character over green seed color. Similarly, round seed is dominant over wrinkled seed. Yellow seed color is controlled by two capital Y genes and green seed color is controlled by two small y genes or factors. Similarly, round shape is controlled by two dominant genes, that is two capital R genes, and wrinkled seed is controlled by two small r genes. According to Mendel, the pure breeding parental plants contain two identical factors for each character. Hence, the genotype of the plant producing round yellow seeds would be capital R, capital Y, and that of plant producing wrinkled green seeds would be small r, small y. Thus, the parents are homozygous for both pairs of factors. At the time of the formation of gametes, each parent produces gametes of only one type. Mendel crossed a true breeding yellow round seed producing pea plant with a true breeding green wrinkled seed producing pea plant. Yellow round variety of plant produces only one type of gametes with a gene content of capital R, capital Y. Similarly, green wrinkled seed producing pea plant produces only one type of gametes with a gene content of small r, small y. The F1 offspring will all be of one variety only, that is plants producing round yellow seeds. The genotype will be capital R, capital Y, small y. It is called a dihybrid because it is a hybrid of two characters and it is heterozygous for both genes. The F1 offspring will bear round yellow seeds because the round nature of the seed is dominant over wrinkled nature, and yellow color is dominant over that is green color. When F1 plants are self-pollinated, that is F1 heterozygous plants are self-pollinated, F1 plants produce four types of gametes with a gene content of capital R, capital Y, or capital R, small y, or small r, capital Y, or small r, small y. The F2 generation of the dihybrid cross yielded not only the two parental phenotypes but also two new phenotypes like round seeds with green cotyledons and wrinkled seeds with yellow cotyledons. It was clear that Mendel observed the segregation of two alleles of a given trait is entirely independent of segregation of alleles of another trait. Thus, a dominant allele of a trait combines not only with the dominant allele of another trait but also with its recessive allele. Similarly, the recessive allele of a trait combines not only with the recessive but also with the dominant allele of another trait, producing four types of gametes with a gene content of capital R, capital Y, capital R, small r, capital Y, and small r, small y. A 16 box checkerboard can be used here to explain the results. Of the 16 combinations, 9 are round and yellow, 3 are round and green, 3 are wrinkled and yellow, and 1 is wrinkled and green. The 9 is to 3 is to 3 is to 1 ratio is a phenotypic ratio. Next, dihybrid test cross. To test the law of independent assortment, Mendel conducted dihybrid test cross, in which F1 plants that is F1 plants were crossed with double recessive plants. The genotype of F1 plant is capital Y, capital R, small r, and that of double recessive parent is small y, small r, small r. The F2 obtained from this cross shows four types of offsprings, that is yellow round, yellow wrinkled, green round, and green wrinkled seeds in 1 is to 1 is to 1 is to 1 ratio. The recessive parent produces only one type of gametes, and F1 hybrid plant produces four types of gametes if independent assortment of these factors takes place. Four types of phenotypes appeared in equal proportions in F2 generation, and this shows that F1 hybrid produced four kinds of gametes by independent assortment of capital Y, small y genes and capital R, small r factors or genes. Next, incomplete dominance. After the discovery of Mendelism, many experiments were conducted on plants and animals to test the validity of Mendelian laws. It became clear that law of dominance has not got universal applicability. In many cases during allelic interaction, it was found that dominance may be incomplete or completely absent. In a few plants, the results of monohybrid and dihybrid crosses obtained could not be interpreted in terms of Mendel's principles. They showed many deviations from the Mendelian inheritance. One such deviation from Mendelian inheritance is incomplete dominance. This was first identified by Karl Scorins of Germany when he was working on the 4 o'clock plant Mirabilis jelpa. Some of these plants bear red flowers, some other plants produce white flowers. The red flowered plants are represented by capital R, and the white flowered plants are represented by small r. When Corins crossed with red flowered plant with a white flowered plant, surprisingly, the plants of F1 progeny produced pink flowers. This is an intermediate expression between two parents. When these F1 plants were selfed, the plants of F2 progeny also showed deviation from Mendelian principles. Three different types of plants were obtained as follows: 25% of red flowered plants, 50% of pink flowered plants, and 25% of white flowered plants. Therefore, red, pink, and white flowered plants were obtained in the ratio of 1:2:1. This is the phenotypic ratio, but according to Mendel, phenotypic ratio should have been 3:1. The genotypic ratio is also 1:2:1. Here, the red color is expressed only in homozygous dominant condition, that is capital R, and white color is expressed only in homozygous recessive condition, that is r. But in heterozygous condition, that is in capital R, the gene for red color fails to express and an intermediate pink flowers are produced. Therefore, this phenomenon is called incomplete dominance or semi-dominance, as called by Gardner et al. In 1991. Incomplete dominance is also known as blended inheritance because it is a mixture of both parents. The appearance of red and white flowers in F2 indicates two important features. Number one, the genes for red and white color did not mix. Number two, they segregated in the F2 generation, indicating that there is no specific gene for pink color. That's all.